Okay, so today we are going to be talking a little bit about genetic engineering and what genetic modification basically means. So my name is Lydia Garas. I completed my PhD at UC Davis, where I worked with genetically engineered organisms. So it's a little bit of a passion for me to try to share what it actually means and try to make it seem a little bit less scary, because there's a lot of money spent on making it sound scary so that you buy non-GMO products. Like, a lot of it is marketing to try to sort of create this sense of fear and drive you into, changing your behavior. But a lot of it is for profit reasons. So just to explain why I'm passionate about it and why I like to talk about it. So we're going to talk specifically about animals. We're not going to spend hardly any time talking about plants, but there are a number of GE plants. Very few of them are available for human consumption, and most of them aren't even marketed in the United States because their value it's not in demand in the United States. So here's a couple things that we'll kind of go over. We'll look at what genetically engineered means and how that's different from a GMO, and if it's different from a GMO or a genetically modified organism. And we're going to look at some of the purposes they could serve or that they do serve. We'll talk a little bit about how, not just my opinion, but how like our collective opinion about GEs or GMOs can actually affect not just ourselves and our own purchasing choices, but it can affect other people. And I'll briefly explain why I talk a lot about diarrhea, because this is one of the main points of my research, and I'm not going to get too nitty-gritty into the details of my research that I did in the past, but I'm still working on some side projects that never quite got finished in my research with GE animals. So what's the difference between GMO and GEs? Basically, a GMO is any living organism that has been genetically altered through any process. So when we think about that, it could include selective breeding. Is anybody here familiar with selective breeding? Couple people? Has anybody ever seen a Pomeranian or like a Chihuahua? And compare that to like a pit bull or like a Great Dane or a Great Pyrenees or a German Shepherd. They look different, right? They're all the same species. All those dogs are exactly the same species, but they have been genetically modified through selective breeding, where humans have decided this dog breeds with this other dog because we want these traits to show up in the puppies. Okay? And we do the same thing with plants as well. We say, this plant made a product that I like, and so did this plant, and so I'm going to fertilize the one with the other, and hopefully the offspring, the next generation, gives me something even better. So humans have been doing this since literally we started domesticating plants and animals. We have been choosing which animals are breeding. So here is the 100 top dogs. You don't need to memorize this slide, don't worry. There's not going to be a quiz on it. But the point I wanted to share you is that these are literally all the same species, and they all came from some ancestral wolf. They all came from the same place. They have literally all the same DNA, but just slight tweaks over time have created these relatively large differences in their behavior and their size and their color and what they're good for, basically. This is Exhibit A. This is Sky Komish, RIP. He he was the derpiest of dogs.
And a bear in this photo.
What was that?
Looks like a bear.
He does, yeah. And he was like 50 pounds, but he was built like a tank, and who knows what he was. He was a whole bunch of different things. But this dog is long since descended from a wolf, okay? But this is an example of a dog who, through tens of thousands of years, we ended up with a dog that it doesn't quite look like a wolf. It doesn't act like a wolf, doesn't behave like them, definitely doesn't sleep like a wolf. Okay, so let's talk a little bit about those GMOs, or genetically modified organisms. So we have directed selection. We were just kind of talking about that We see it a lot in plants. We see it a lot in animals like dogs, but we also see it in things like cattle and goats and sheep and any other domesticated animal you could think of. So that's one way to do it. The other way to do it is what's called mutagenesis. And this means to create mutations. That's all that means. There's a bunch of different ways that we can create mutations. One of them is through ionizing radiation. We literally just blast things with radiation, and that damages the DNA. And when the DNA tries to repair itself, it makes mistakes. And those mutations could potentially be heritable. Chemical mutagens do the same thing. It's just a chemical instead of ionizing radiation. It's still damaging the DNA. When the DNA tries to fix itself, it makes some mistakes in the process of fixing itself. And so we get basically a new sequence of DNA, or a slightly altered sequence. It's not like a brand new sequence. It's just tiny little edits, like a typo. Think if you're writing something or you're doing voice to text and you see, oh, that's not exactly what I meant. And the body's usually pretty good, and cells are usually pretty good at fixing mutations, but it doesn't fix every single one. So humans learned this. So we actually will radiate and expose to chemicals plants a lot of the time. That's kind of where we started. That was the training grounds for playing with DNA before we even knew too much about what was going on. So that's another way we can do it. Another thing we can do is called induced polyploidy. And this is basically where we force organisms to have multiple copies of the same gene, sort of like over and over and over again. The interesting thing is, this can occur naturally as well. So some organisms just have too many sets of chromosomes, basically. And those oftentimes tend to be more robust and have larger fruit. And interesting, most seedless fruit have polyploidy, whether it's induced or naturally occurring. So think about your favorite seedless fruit.
Lemon.
Okay, yeah, there you go. Lemons, watermelon. Most of those plants are going to have polyploidy. That just means they have too many copies of their DNA. But again, it's to our benefit. That's also one of the reasons why we can't just grow some seeds. Like sometimes the plant will make a rogue seed every once in a while, but those seeds don't always germinate. I just saw a comment in the chat that they are all good dogs. They are all good dogs. So these are the sort of like old school ways of doing things. We still do them, to be clear, but we now have more refined ways to make changes to DNA. One of those is through transgenic modification, and the other one is through gene editing. So again, what I'm trying to get at is we've been playing with DNA for a long time. We've used some mechanisms that are sort of what we call a shotgun approach. We're just creating mutations, crossing our fingers, and hoping something good comes out of that mutation. When we look at the more recent technology, these are really targeted, precise, directed changes. So we are going in, we say we want to change this gene, add this gene, take this gene out, make this exact mutation. So it's a lot more controlled, actually, than what we've done historically. So transgenic modification is what we would consider to be genetically engineered. And gene editing is still so new that there's debate if we should, and by new, I mean 20 years. I'm not talking about yesterday. Should it be considered the same as genetic engineering? Is it a subset of genetic engineering? Do we want to lump it together? Are people going to be afraid if we combine them and call them, oh yeah, it's just a different way to do the same thing? They're all just different ways to do the same thing. Some ways are more efficient, some ways are less efficient. But we don't call gene editing GE because we already use GE, even though it would make sense. So when it comes to the term GE or genetic engineering, you guys have probably heard the phrase, a square is a rectangle, but a rectangle is not a square, right? So something that has, 4 90-degree angles, but a rectangle can have, 2 long sides and 2 short sides, but a square always has 4 identical length sides, yeah? So when we talk about GMOs, we're talking about all things where we've played with the DNA. When we talk about GEs, we're just talking about a subset of animals who've had DNA altered in a specific type of way. So again, all types of DNA alterations, GMO, subset of those types of DNA alterations would be GE. And this includes a couple of different techniques. So I briefly mentioned we can mess with the DNA through this shotgun approach of radiating or exposing to chemicals. But when we talk about genetic engineering, we're talking about directed changes to the DNA. We can do that through cloning. We can do that through transgenic modification. And this is usually used to insert a gene. Usually. There's a variety of ways that you can do this. And then there's gene editing, which again, it can be used to insert genes. It can be used to delete genes. It can be used to modify them. They're already there, but we can sort of make small changes to those genes. And sometimes those small changes are one single nucleotide, one single molecule different. And we're going to talk about briefly, just very briefly, I'm going to give you a crash course on what DNA is, so we're all on the same page. Okay, so what is DNA? Raise your hand if you've taken any kind of biology class, like ever, anytime in your life. Most folks, okay. Even in high school, like that's totally fine. Maybe you learned about it in 7th grade, it's been a long time, and yeah, you learned it, but you probably forgot it. That's fine. The fundamentals of DNA are that we use 4 molecules, A, T, G, and C. That's it. Every living thing on this planet uses A, T, G, and C. That includes slugs, that includes plants, that includes humans, that includes dogs, that includes monkeys, that includes squirrels, it includes fish, salamanders, insects, everything uses A, T, G, and C. Now, the interesting part is the way that you order the A, T, G, and C is what gives you genes. Not blue genes, but like the genes in our DNA. So if you have one sequence, that gives you one gene. If you have a different sequence, that could give you a different gene. Okay? When we talk about biology in general, we sometimes refer to the central dogma. It is this overarching theme of how all life makes proteins. And we have to start with our DNA. This is where we find the A's, T's, G's, and C's. And then we go through a process called transcription. There's enzymes involved. Basically, it reads the DNA and it makes a similar but slightly different RNA version of that. Okay? This RNA, again, similar molecules to DNA. We use A, U, G, and C instead of A, T, G, and C. Basically the same type of structure. Instead of having double-stranded molecules, we have single-stranded molecules. And then these single-stranded molecules, this is in the nucleus, this is made in the nucleus of a cell, this gets sent out of the nucleus of a cell, and that's where ribosomes turn, or they read the RNA and translate it into protein. And that's a process called translation. So we basically take our genes and our DNA, we make an RNA copy so we can get it out of the nucleus where it can be made into a protein. Every animal, every plant, every living thing on this planet does it this way. So not all animals have the exact same genes, but yes, all animals use the same 4 ATGC bases, which base pair. So how close are we to other organisms? I just picked a couple of things. 65% of our DNA as a human is the same as a chicken. I don't know if you've met a chicken recently. They seem pretty simple. They're pretty cool though. But they've got a lot of really similar structures to us. If you think about it, they make muscles, they got a brain, they got bones, they got legs, they've got arms, technically, their wings. They've got a liver, they got a digestive system, they got a mouth, they have eyeballs just like we do. We are 88% similar to dogs. Getting a little closer. Sometimes they say people resemble their dogs. It's not just by appearances, right? And we are 98.6% 98% similar to chimpanzees. That means we share 98.6 of our genes, percent of our genes with chimpanzees, approximately. And again, slight variations. We have some genes they don't. They have some genes we don't. And we have just tiny little tweaks to those A's, T's, G's, and C's in some of the other genes that we have. But largely speaking, a lot of animals, we just share so much of our DNA. So now let's talk a little bit about different ways to make GEs. And I don't want you to try to memorize again. And I know you're here to learn about GEs and GMOs. There's, again, not going to be a quiz on exactly how this works. But have you guys heard of Dolly the sheep? Also RIP. She is in a museum right now. Basically, the way Dolly the sheep was created is they took an egg that has a nucleus inside it, and they took the nucleus out. That's the process of enucleation. So they took the nucleus out, which means they took all the DNA out too. Then they took a cell from the mammary gland of a different sheep, and they took the nucleus out of this cell, and they put it into the egg. They zapped it with current, literally they zapped it. And then it started to divide like an embryo normally would divide. They transplanted that embryo into a surrogate, and that embryo became Dolly. She was born as a lamb. She grew up like any other lamb. And this is an image of her. This takes a number of animals. It's quite time consuming. You have to wait for the animals to go through multiple reproductive cycles. Not always 100, it's definitely not 100% efficient, okay? So not every embryo that you create is going to survive. Not everyone is going to divide. Not everyone you transplant is going to actually grow. Not every animal is going to be born alive. There's natural attrition that happens regardless, but it's time consuming and it's very expensive. So that's one way we can do it. We are making an identical copy of this animal because we're taking this animal's DNA and we're basically making a brand new embryo out of it. So this animal should be genetically identical to Dolly because all the DNA came from this mammary cell. Yeah.
Why did they have to use the mammary cell? Couldn't they have used any other cell?
They probably could have used any other cell. Some cells work better for this process than other cells. So it's possible that they had some growing in culture and it was what they had on hand. It's possible that that they had done it before with other types of cells and they found that that one worked better than the other types of cells. We sometimes also do this with stem cells. Yep.
Because they're so adaptable.
They are, exactly. So the nucleus will have, it will have been less committed, let's put it that way, to a certain pathway that it was already going to go down. Yeah. So it probably didn't have to be a mammary cell. But oftentimes we use skin cells for this type of process because they're so easy to come by. We have so many of them. And we also have stem cells in our skin that could potentially be like not just the dead skin cells on the surface. We wouldn't use those ones. Okay, so let's talk about some other ways to make a GE organism. One of those ways, cloning. Another way is using a viral vector. And a vector is just a thing to take DNA from point A to point B. So if we have a virus and we alter the virus and we include a piece of DNA in that virus that we want to now get into our cell type, whatever that cell type is that we're interested in altering, we use the virus to infect the other cell, okay? But these viruses have been modified, so they're not going to cause disease, okay? They're not going to cause like a symptomatic disease. We use viruses for a lot of different applications in biology, and it's literally just used as like a shell, a shuttle, if you will. It's not a pathogenic virus. It's not going to make you sick, okay? And this can include permanent changes to chromosomal DNA. So basically, there's a diagram right here that you can see. We've got a patient right here, so they've got a disease. We can take some of their cells out. Say the cell has this gene in it, and say this gene is damaged, it's not functioning properly. So this person has some sort of genetic mutation. Maybe it's causing a debilitating disease, right? So then if we have these viral vectors that carry a fixed copy of the DNA, the one that the rest of us all carry, it could potentially integrate, basically cutting out this broken piece of DNA, the broken gene, replace it with a properly functioning gene, and then you take those cells, the virus will eventually eliminate itself, it will die off. You take those cells and you inject them back into the patient. And now the patient has a subset of cells that can make the correct protein instead of the incorrect protein. We do this already.
Is that like CAR T therapy?
Yes,
Kind of close.
close. It is. It depends on what type of, if we're using T cells, immune cells to do this, but yes, we can also do that. And this is sometimes called an autologous transplant, where you take your own cells and put them back in your body after you've edited them. But we are now seeing, I will talk a little bit about it, but like off the shelf, which means it doesn't have to come from the same person who's sick. So it could potentially come from a donor. Again, we won't get deep into those weeds, but it is one way that we can edit out a sort of broken gene and replace it with a fixed copy of a gene.
And once these cells are inserted into the patient, they live in perpetuity. I mean, they would start to reproduce themselves, or do they have a lifespan?
Good question. So in an ideal situation, they would live in perpetuity, but they would not grow uncontrollably, because then you have cancer, right? So you would want it to be sort of a finite amount of cells that could replace or maintain a certain level of protein production without growing uncontrollably and creating a different type of disease in that situation. Okay. So what I showed you here is the ex vivo. Ex vivo means outside of the living body. So you take the cells out, you play with them outside, and then you put them back in. In vivo means inside your body, in the living organism. And that's where you would directly inject the virus into the person's body to alter the cells in the person's body instead of in a petri dish, basically. Okay, so that's another way we can do it. So we got cloning, and we've got using some sort of altered virus, viral vectors. Another way we can do it is microinjection, where we literally take, this is an egg, or I can't quite tell if there's a pronucleus. Could be a fertilized egg, so like a single cell, like it hasn't started dividing yet. And in this case, you take this tiny thin glass needle and you poke it through the cell, because at this point it's just a single cell, and you inject it with the gene of interest. And sometimes there's additional enzymes to help it, or some additional instructions to help it find the right place to do the repair, or to insert a specific piece of DNA that you want to insert it. Now, oftentimes in the past, we had to cross our fingers again and hope that the gene that we were putting in, say it's a new gene, or a corrected, fixed version of a broken gene, we didn't know exactly where it was going to go. It could have gone anywhere on any chromosome. And that could mean going into an important gene and then breaking that other important gene, and then this embryo doesn't make it, right? So we don't do this with humans, right? This is, again, kind of like an older way, one of the OG ways of doing microinjection was just inject the DNA, cross your fingers, hope it goes someplace that's not bad. And also hope it goes someplace that's not just going to be silenced over time, because there are parts of our DNA that are expressed throughout our lifetime, and other parts of our DNA that are only expressed for short periods of time, and then they kind of get shut down. Yep, exactly. So we've got some epigenetics in there. So we can silence some areas of our DNA, and you don't know where it's going to go in that situation. But this is one way to take a new gene and insert it into an existing chromosome. So when we talk about a transgenic organism, so we've talked about GMOs, we've talked about genetic engineering. A transgenic organism is an organism that has a gene from a different animal, a different species. So this gene maybe comes from a different species than whatever this is. Okay, that's just one subset of sort of outcomes that you might be trying to do, or you might be trying to create. Next up, raise your hand if you've heard of CRISPR-Cas9. CRISPR technology. It's a little bit of a newer, but also it's been relatively a hot topic in the past few years. This is a massive development on just microinjection, crossing your fingers and hoping it goes where you want it to, or someplace not bad. This helps us control where to make the edit. So when you microinject this complex, and again, we're not going to get too deep into it, you basically can direct where to insert the gene. I want it to go right here. I know this is the gene I'm trying to disrupt. This is the gene I'm trying to cut out. This is the gene that's damaged and I'm trying to fix it, and I want it to go right here. So this uses an enzyme and what's called guide RNAs. Remember RNA? It's just a single-stranded sequence of nucleotides that lines up to the section of the chromosome where the gene is that we care about. And it tells it, go right here. So if this piece lines up with this piece, the enzyme comes in, it makes a little nip in the DNA, puts your new DNA in that you've put in there. Or it can make just a single base pair change. Say there's supposed to be an A in this location, but you've got a mutation in your DNA and you have a C there instead, so your gene's not working properly. It can go in there and say, switch it back to an A. We can make it that tiny of a change using this technology. So think about the possibilities. One of the biggest ways that this could impact humans specifically is by providing a potential way to cure genes, genetic mutations, and diseases that are caused by like one single gene. So a mutation in one single gene can cause a, catastrophic disease. But if we can fix that, potentially, we might be able to cure it. And I don't mean treat the disease, I mean fix it. I mean change the DNA so you no longer have that broken, mutated copy, that mutated version of the gene. Now, that said, if there's already been extensive damage to your body, that's not great. But if we could fix it from getting worse, we could stop it from getting worse, that would be fantastic.
And if you could repair the pre-existing damage through refined use of stem cells.
Yes, and that's the thing, is that so once you, and it depends on what kind of disease we're talking about, what kind of damage we're talking about, but yes, absolutely. Absolutely. Yeah. Okay. So it's actually being used already. And it's actually being used to treat incurable diseases, hopefully formally incurable diseases. So this is from the Daily Mail, so please excuse the clickbait headline on this. But 2 babies with terminal cancer are cured after miracle treatment. Couple things I wanted to point out. This is 2017. Okay, we're already talking about 7 years ago. This is being done. Not brand new technology. 2 infants, basically, who had leukemia. They had already gone through treatments like chemotherapy. They'd already gone through bone marrow transplants. This is like, we have no other option. Our kids are going to die if we do nothing. Sure, let's try this, okay? That's desperation, right? If you've never been in a situation where you have that kind of desperation, consider yourself lucky, right? However, could potentially have long-lasting impacts. This is just 2 children. If it became available to other people who had leukemia, that would be even better. October 2022, getting warmer, right? A little closer to where we are right now. This is where we get into that CAR T cell therapy that you had mentioned. So they used donor T cells. They genetically engineered T cells from donors, not from the children themselves, but from a donor. And again, they treated these children who had incurable leukemia. They'd already tried every other possible therapy. That was groundbreaking news that it could happen. And then it did. Just over a year ago, 2023. Another child using genome editing. And again, targeting leukemia, which is a type of blood cancer. So when we think about these things, oftentimes it's like, okay, it's big and scary and I don't understand it and I'm never going to need it and I don't want to use it. So like, maybe we shouldn't, right? But then when you realize there's people who have like life or death choices to make. And this is their choice, right? They could have said, you know what, we don't want to pursue this treatment, okay? But they don't stop other people from pursuing the treatment. And in this case, it worked, okay? And again, we're talking 2023, relatively recent. If it buys a couple years, that's better than 0 years, okay? If it buys a couple months, it could just be better than anything else. Like it's better than the alternative, which is resigning yourself to the fact that there's nothing you can do about it. But I do want to point out, it's not just what could we do, it is what can we do right now? What are we doing right now? Okay, so we talked a little bit about why this is kind of cool, why it's interesting, how it can be worked into actually treating human diseases. Why would we do this to animals? Why don't we just stick with our inbreeding our animals or selectively breeding our animals and our plants? Well, that takes a lot of generations. It takes a lot of time. And again, it's imprecise. It's, you know, let's hope that if we breed these two animals or these two plants together, we get something that we want out of it. And maybe we do and maybe we don't. But when we make genetic edits to animals, we usually have a couple of goals in mind. One of those goals could be increased growth. So you can have higher yield. And when I talk about yields in animal production, my background is in animal agriculture. That means we have better milk. That means we have better muscle. That means we have more milk or more muscle. And muscle is meat. Like that's protein that's for consumption. We could also engineer them to be disease resistant. And we'll talk about a couple examples. Animals have been engineered to have less of an environmental impact so that farming the animals does not have the same sort of catastrophic impact on the environment that traditional farming methods oftentimes do. It can improve animal welfare. And even if you're a person who's omnivorous and you eat plants and animals, I think most of us can agree that we would rather have healthy animals that are well taken care of than to not have them be well cared for. They can also be used for, again, more direct medical applications. So we can use them as medical models to study diseases that are found in humans. So we can sort of recreate a disease like it would be in a human, and then we can study it in an animal because it's really hard to study things in people. It's not just expensive, it's sometimes impossible to study things in humans. So we have to study it in something else. We can use them as bioreactors. And what that means is they can produce a large volume of something that we can purify, that we can sort of isolate from them. And then the last one is xenotransplantation. And xenotransplantation, you guys all know what a transplant is, right? You take an organ from one human and put it in another human.
Like you grow an organ in an animal first.
Exactly. That's where the xeno part comes from. You're taking it from a different animal and putting it into a human.
It's the effort that's trying to be done.
Absolutely. That's one of the things we're gonna get to. Brand new. Not just heart valves. Heart valves we've been doing for a long time because they don't have a lot of our body doesn't necessarily recognize the components of a heart valve as being foreign. So we've been using like pig heart valves for a very long time, but we'll get to it. No spoilers. So let's talk a little bit about some examples of increased growth. Has anybody heard of Aqua Advantage salmon or Aqua Bounty salmon? So this company, I think, has been around for 30 plus years. And I feel so sad for them because they have gone bankrupt almost more times than I can count. And they have always just come back from the cusp because they're like, no, really, we think that this is going to be amazing in terms of it basically increasing the rate that salmon reach market weight. So here's a graph from the days from first feeding. So the first time they're feeding them food from after they hatch, and their weight in kilograms. And if you notice, standard salmon, they take a longer time to get to the same weight. So if we're just looking, even just comparing this like 4 kilogram mark right here. And the Aqua Advantage salmon, they basically grow faster up to a certain point. I'll show you another graph on the next page. This is a standard Atlantic salmon that's the same age as this genetically modified Aqua Advantage salmon. And technically, this is not just a GMO, this is a transgenic, it's a genetically engineered organism. So they're the same age. This one looks much bigger, right, than this one. That means this one can be eaten sooner. So if you're here for healthy protein, this animal is going to reach market weight and be usable for human food much sooner. And basically the way that this worked is they took genes, a growth hormone from a Chinook salmon, so still salmon, but it's a different species of salmon. And that accelerates the growth a little bit. And they took a piece of DNA from ocean pout, which is a different type of fish, and that helps turn on that Chinook salmon gene. So they took 2 little fragments of DNA from 2 different fish and then put it into Atlantic salmon. And now those fish can grow year round. Normally, standard salmon kind of stop growing in cooler months because the temperature turns off that gene that allows them to grow. So when it's cold, you don't eat as much, you don't grow as much. Well, now that gene is on year round, so they just grow year round. But the interesting part is everybody was like, well, are they going to become these gigantic, like enormous, like shark-sized fish? False. No, because every other gene in their body is still an Atlantic salmon gene. It's still a salmon gene. So you can see at the end of this graph here, they're basically the same exact weight. So by the time they reach 4 years of age, they're the same exact weight. They just got there a little faster. So the blue line is the genetically engineered. And they reach that market weight in about half the time. So instead of having to farm them for 3 whole years before you can sell them, you can farm them for a year and a half before you can sell them. And that has less of an environmental impact as well, because then you're not growing an animal for 36 months for 3 years, you're growing it for 18 months for just a year and a half. And the reason that this is so important has to do with feed conversion rate. And just a quick primer on animals. When you feed them something, a lot of it comes out of their body as waste, okay? If you feed a cow or cattle 8 pounds of food, their body will gain 1 pound. So that means 7 pounds is just, it's wasted, it's lost, okay? It helps them keep their body warm, helps them move around and do other biological systems, but you're losing a lot in the middle. Pigs are better. You give them 3 pounds, they gain 1 pound. Much better conversion rate. Chickens, even better. You give them 2 pounds of food, they'll gain 1 pound or give you 1 pound of eggs, for example. Fish is almost 1 to 1. They have almost no weight loss. They're what you would think of like them defecating, like pooping out, is much, much less. They're not wasting energy trying to keep their body warm. like the rest of these organisms are trying to keep their bodies warm. So if you give a pound of food, you get almost a pound of meat from these animals, or a pound of like mass in their body. So this is one of the reasons why fish is considered one of the most sustainable sources of animal protein, because you get out almost what you put in, or it's much closer. It's not exactly 1-to-1, but it's very close. The FDA approved in 2015 these salmon to be consumed after years and years and years and years and years and years and years of discussion. And we need more data. Here's more data. We need more experiments. Here's more experiments. We need more this. We need more of that. And they kept doing it, and they kept doing it, and they kept doing it. Finally, the FDA said, like, okay, you're right. There's a, sure, sell them. Yeah, people love to hate the FDA. And also, has anybody ever seen Aqua Advantage salmon at the grocery store? That's because a lot of the major retailers, Walmart, Costco, Target, Safeway, Fred Meyer, swore they would never sell genetically engineered salmon. Because customers, they were afraid their customers would be afraid to eat it. What do you think that sent the message to the customers? Do you think that tells the customers it's safe to eat, or do you think that tells the customers you probably shouldn't eat this?
It becomes a self-perpetuating cycle.
It does. Basically, if you say, oh, I swear we're never going to sell this thing,
Where could they get Aqua Advantage salmon and walk?
you can't. They literally, they're working with like distributors because of the amount of fear just around genetic engineering is that, and because none of the major grocery stores, like the retailers that you and I would normally shop at, they all were like, we commit, we're never going to sell this product. And I'm out here like, man, but I want to be sustainable. Like, I want to be environmentally friendly. I want to eat this salmon. I'm not afraid of GEs. How do I buy it for myself? And it's like, well, sucks to be you. So their initial sales, even after they were approved in 2015, their first sales didn't really happen until 6 years after that. That was when they found a buyer. And again, this company has been around for decades, and they've been trying so hard to prove, like, every request that the FDA has sent to them, they have come back with data that says, here's what you asked for. Here's the evidence that you wanted that shows this is sustainable. There's no risk of these animals escaping into the wild. They're cultured in Indiana right now, like nowhere near a river that the water could potentially leak into and release a bunch of Atlantic salmon. They're all female, sterile females, so they can't even reproduce if they try. So they've done all of these sort of backup measures to try to ensure that those potential consequences are like impossible or very unlikely to happen. There's some questions in the chat. Let's see. How healthy is it compared to other salmon? It's basically the same as other farm salmon. So in terms of like their, the omega-3 fatty acid, like the same, we're talking very similar in makeup to other farm salmon. And isn't this fear based on the unknown long-term impacts on human health? It is based off of the fear of unknown long-term impacts, but if you eat Chinook salmon, or you eat pout, or you eat Atlantic salmon, and we know that all DNA is just DNA, there's no scientific basis to be afraid of any long-term impact on human health.
I think that's really funny. The same people who are caring about this impact are the same people who aren't caring about the impact that all the beef that they're eating is having.
There you go.
All the pollution that it's causing, that it's actually having on ourselves.
There's definitely a lot of hypocrisy when it comes to like, I want to do this because this is good, but I don't want to do this because this is bad. When maybe we don't understand all those things and it's just a gut reaction. But I do want to be clear though, fear of the unknown is what keeps animals alive, including humans. You don't go outside and eat a random mushroom that you see, right? Because you don't know what it's going to do to you. You wait until somebody else eats that mushroom or another animal eats that mushroom and you watch them. You watch what happens to that animal. You see, do they like hallucinate for a week? Do they die? So that fear it's literally ingrained in our, not just human DNA, but in any animal's survival, is to be a little bit suspicious of anything new. Maybe a lot suspicious of anything new. Luckily, we understand how DNA works. So we can sometimes, if we try hard, override those fears with knowledge. We can override it with some common sense and understanding and say, okay, this is not going to be, if anything, the human health impact is going to be, maybe this salmon will be cheaper and I'll get more omega-3s in my diet and we'll have less environmental pollution because I'm eating salmon instead of cattle.
I saw how big that song was. Everybody will eat that.
I mean, someday, right?
It's
Okay.
interesting that the people who are so into sustainability
Mhm.
still find this unacceptable.
Well, they don't,
Sustainability on one hand, not on the other.
and I do want to point out, this is not just unique to genetic engineering. This is sustainability also has been very closely associated, not for good reason, but very closely associated with organic farming. And organic farming is one of the least sustainable practices. So do not be fooled. If you want to be sustainable, that's different than just going out and buying something organic. Okay? They do not mean the same thing.
All those recipes that you must have organic eggs are just, I don't believe in it.
I mean, why are they telling you it must be something or something else?
Oh, maybe it's because I looked where the book's author is located, what they're all about. Oh, wow, looks like they're very anti-GMO.
That's the thing. A lot of time, if somebody tells you you should feel this way or you must do something in a specific way, look to see if they have affiliate links on their website.
Oh, no, it's
Look
hilarious.
to see if they're getting some sort of kickback from some sort of company. If they get,
so funny.
if you click on their links, if they get something back, definitely that's a good, we talk about that as like science literacy, like making sure that when you see something, maybe we don't believe everything we read the very
Yeah,
first
like an
time.
official book, and you go look at the author's page and you realize that they're getting funded by the programs that they're saying
Oh, yeah.
they're good or bad.
My favorite is an article that was published that said 100% of ground beef contains fecal bacteria. It was published by like veganhealthnews.com. And I'm like, I don't know if I trust vegan websites to have academic scientific rigor to test a meat product. They may have an ulterior motive. So I'm going to go find a different article. Like, sure, keep that in your mind, but go find an article from maybe a more reputable source, right? Okay, good questions. So how else can we increase yield? We've done this in pigs as well. So some pigs have been genetically engineered to produce more milk. And when they produce more milk, that means their offspring survive, more of their offspring survive, and they're larger when they're weaned. And usually pigs are weaned around 3 or 4 weeks. That's kind of when they're like done with their mom. And this isn't just about making more pork for humans to eat. This is also an animal welfare issue because if the offspring survive, that's better for the offspring, right? We don't want them to die from malnutrition because they don't get enough food. So again, sometimes when a scientist goes out there and says like, I want to genetically engineer an animal to do X, Y, Z, it's like, oh, as a side effect, we get this sort of additional bonus. Like we get increased survival, increased weight. It's not just about increased pork. It's also, oh, this is actually better for the piglets as well. When it comes to changing the milk and the muscle composition, so not the amount necessarily, but the makeup. The majority of saturated fats in our human diet come from eating some sort of ruminant meat or milk product. So that includes cattle, goat, and sheep, or it comes from pork. Okay, that's the vast majority of our saturated fat. Unless you go all in on like coconut oil and avocado. That's like the only other like major sources of saturated fat you might have in your diet. So if we genetically engineer animals to make leaner meat, so have less fat in the meat, or have less saturated fat in the meat, or have lower fat in the milk, or have a longer shelf life. So it literally will last longer in your refrigerator before turning green and smelling really bad. We could and have engineered allergen-free milk. So there are some major components of milk. There's two major proteins, and I'm not going to get into the weeds, but the folks who are allergic to like dairy milk oftentimes are allergic to a specific protein. So if we can get rid of that protein so they don't make it in their milk, that means that that could be a source of nutrition. And also sheep have it genetically engineered to have finer wool. So longer fiber, thinner fiber, softer fiber, increased fiber.
Could be, could cheapen the prices of clothes.
It could, absolutely. And especially when you consider how you've probably heard of like microplastics and how a lot of our synthetic fibers are actually, they're ending up in places we don't want them to end up, like microfleece, that kind of stuff. Yeah, so if we can use wool, that could potentially be more sustainable and less environmentally damaging because it's a biodegradable product, but wool also lasts a very long time. Okay, let's talk about bioreactors. So one of the very first approved animal products that was approved for human use was an anticoagulant protein. Now, anticoagulants are used oftentimes if you have stroke or blood clots, and you sometimes get put on anticoagulants for, a short term, and sometimes it's the rest of your life. So the way this works is these organisms, these goats, were genetically engineered to make this protein, which is called antithrombin, in their milk when they lactate. So only the females make it, and it's only in their milk. And don't worry, this is not the goat milk that you see at Trader Joe's. Okay, these are like medical grade goats. Okay, they live separate.
There's a sentence I never thought I'd hear in my life.
A medical grade goat.
A medical grade
Yeah.
goat.
Yep. Trust me, their lives are very, very well protected. So this was first approved in 2013. And that was because we literally could not get enough antithrombin to treat everybody who needed antithrombin. And somebody was like, well, what if we just have goats make it in their milk? And then we just purify it from their milk. And it was like, oh yeah, we could do that. So they inserted a gene for a protein. The protein codes for antithrombin. They milk the goats. They purify the protein out. They make sure it's properly filtered. And then it is a safe injectable drug. I also, I included a link to spider silk in case you're interested in learning a little bit about goat's milk that creates spider silk protein. Spider silk is one of the strongest natural fibers on this planet. And it's somewhere between, it's probably around 5 minutes long. So if you guys are interested, I'm not going to play it right now, but it is actually quite fascinating to see. The hardest part was to try to figure out how do we like spin it into a fiber, basically. And one aside, insulin for diabetics has been produced by genetically engineered E. coli since 1986. Most people probably don't think about that. We used to just squeeze it out of pig pancreases. That's not efficient. There's a risk, right? Like the pigs were just going to be slaughtered for meat anyway, so we would just extract the protein from it. Now we have a much easier way. We can make a lot more.
Doesn't mean it's not still overpriced.
It's way overpriced because E. coli, I mean, yes, there's definitely some issues with keeping it clean, keeping it from getting contaminated with anything. But since 1986, we have been using genetic engineering to benefit human health, and probably even before that. Milk composition in bioreactors continued. We have other types of organisms, cattle and goats, that are producing two different types of antimicrobial enzymes. These are my faves. They're the ones I worked with on my PhD. And those antimicrobial proteins are found in human milk. And they basically just said, we'll take this gene and we'll have goats express that gene in their milk. Not the rest of their bodies, just in their mammary glands, just when they're lactating. Couple other things, improved animal welfare. So we mentioned like the piglet survival, that's good for the piglets. But unless you've had mastitis or seen an animal that has mastitis, probably not super familiar with it. It's one of the leading causes of disease in dairy animals. So it's an infection in the mammary gland, and dairy goats, dairy cattle don't produce those antimicrobials in their mammary glands like humans do. But if they're genetically engineered to do so, there's a potential that the antimicrobial will sort of inhibit bacterial infections, which will then decrease the chance of infection in the mammary gland, and it will decrease the need for antibiotics. Because if you don't have an infection, you don't need to treat it with antibiotics. How are we doing on time? Okay, we still got 10 minutes, right? Ish. Okay, more welfare stuff. Most cattle and goats and sheep have horns, and horns can be very dangerous, both to the animals, to themselves. They get stuck in fences. They can be dangerous to other animals, and it can be dangerous to the people working with them. So most cattle, sheep, and goats are disbudded. And what that means is before their horns even start to form, they take a red hot iron and just burn it, cauterizing it. They're killing off the cells that will form a horn. Then it doesn't grow a horn. This is different than dehorning. Dehorning is when you have a horn already and you have to take the horn out, and that's way worse.
X wants to get infections.
What was that?
X wants to get infections and stuff.
They can. I think the bigger risk is actually just injury, like self-injury, injury to other animals. But the interesting thing is red Angus beef cattle, they're literally just beef cattle that are red instead of black, like the black Angus beef cattle. They have a mutation in the gene that forms horns. It's naturally occurring. And people were like, well, what if we just had that mutation, but we had it in dairy cattle? Then dairy cattle wouldn't make horns either. And then we don't have to do this painful process of disbudding, and then none of them make horns. The problem is, historically, the only way to do that is to breed a beef cattle with a dairy cattle. But then that would set the industry back like 100 years, because dairy cattle have been bred to make milk, and beef cattle have been bred to make meat. And if you start crossbreeding them, you're going to get cattle that aren't good at either. Okay, not good at making milk, not good at making meat. And then you have to breed and breed and breed and breed for generations. We're talking, like I said, 100 years before you could create basically all new dairy cattle breeds that have this Angus mutation in them, but no other Angus traits, because we don't want beef cattle traits in them. So instead, they did targeted gene editing. So again, gene editing, where they basically create the same mutation that's in the Angus cattle, they create it in these dairy bulls. They're still young, little babies. But this is them. This is two bulls that have one mutation, like just one gene mutation, but it's the same as a beef cow or beef cattle.
And those are both red Angus? I've never even heard of red Angus. Isn't that?
These are Holstein dairy cattle. No,
Angus.
this is actually probably a Jersey. I'm not 100% sure. It's a little too close up, but there's nothing in them that's anything beef cattle, except scientists knew which gene, which mutation, and they recreated that same gene's mutation because they have the gene for horns. These guys, the red Angus, have the gene for horns, but it's mutated so they don't grow their horns. Gene's still there, just broken. Basically, they just broke the same gene here that was already broken in the other animal. So now they don't produce horns. So these bulls can now be used to breed, and basically without losing 100 years, can create purebred Holstein cattle that don't form horns. It'll take a couple generations to build up the numbers, but not 100 years. Yeah.
Is it possible to reverse that? Because like, what if we can, breed cows to where, oh, we don't have horns for them. Well, now it's been like 20 years or so, and now we can't have any of them horns. Like none of them are growing horns. Is there ways to reverse that so they can grow horns, or is that a work in process?
I don't see a world where you would want to do that, where you'd want to reverse that. But in theory, you could just go back in and edit that same gene and put in the gene that's not mutated.
If anybody's ever seen two longhorn bulls fighting,
Oh,
believe me,
it's
that
violent.
painful cauterization is preferable.
Yes. Yes. This is This is preferable to animals goring each other and causing fatal injuries to each other or to the humans that work with them. This is even more humane. Decreased environmental impact. So this is another way that we can sort of, in addition to the, farmed salmon, if they grow faster, we can get them to the market faster. In both animal and plant agriculture, there's a lot of waste runoff. It's usually from nitrogen and phosphorus. We put it on the fields, we put it in the food, they grow, but then there's waste and it runs off into our waterways. And this can cause what's called eutrophication. And this is where you end up having so many nutrients in the water, the algae bloom, it blocks sunlight, everything dies, it becomes a swampy, gross mess. Okay, that's not good for the ecosystem. That's really bad. So what some folks did was they engineered pigs to create this enzyme to help break down phosphorus in their mouth. So they could give them less phosphorus because they can use the phosphorus that's in their food without having to dump a whole bunch of extra phosphorus in their diet. Because if you put it in a normal pig's mouth, that extra phosphorus is going to come out the other end. And that's when you get these sort of swampy, again, green algal blooms. So again, it's cheaper because you're not giving the pigs as much phosphorus and it's less environmentally damaging. Disease resistance. Have you guys heard of avian influenza? It's been back in the news lately. Well, there have been chickens genetically engineered to resist avian influenza. When the last sort of wave of avian influenza came around, decades ago, we had to kill off like millions of chickens to stop the spread so that we didn't risk having the birds spread it to wildlife or to humans because it can be very dangerous to humans. There's also cattle that have been engineered to resist developing mad cow disease. This is less of an issue, but proof of principle, we can engineer animals that resist specific diseases that could be fatal to humans as well. There's a couple different ones, pigs that are resistant to swine flu, also good. And we have GE mosquitoes now too. And it's not so much that they're disease resistant, it's that they die off and they don't transfer the disease to humans. So think like dengue, West Nile virus, Zika virus. Mosquitoes are a huge source of human infection. So they have genetically engineered mosquitoes. And they started releasing them actually just a couple years ago.
The anthrax epidemics that just wiped out huge herds in the late 19th century.
Yep. I don't know that there's any organisms resistant to anthrax in particular, but they might have a vaccine, which
They
maybe
did have vaccines back then, but I'm wondering if genetic engineering's been used to get rid of the need for vaccines. They did develop a vaccine then.
I was going to say potentially, but if there's already a solution, creating a brand new line of organisms might be not the most cost effective. And then there's xenotransplantation. I think this is probably what we'll get through at the very end. So basically, this is where we use a different species as an organ donor, and the human recipients. If you just took like a random animal's organ and put it in a human, your body would recognize it as being foreign and destroy it immediately, and then you would die because you need that organ in order to function. So there's a couple things you have to do in order to make xenotransplantation even feasible, like potentially work. And one of those major things is each species has specific cell markers on it. So humans have like human specific, pigs have pig specific, cattle have cattle specific, goats have goat specific. But there's also human specific markers. So it's not enough to remove the animal cell markers. You have to put the human cell markers on there as well, so that the organ that you transplant is recognized as being, oh, this is okay. It's not just that it's dangerous. It's that, oh yeah, this is fine. This is also human. So it requires gene editing, and you can do this in a variety of different ways that we've talked about it. So this is what it looks like to develop a GE pig for gene editing. This is one of my former interns. Her name's Grace. She learned how to artificially inseminate pigs a couple years ago on her spring break. We do different things for spring break when we're scientists. The embryos are flushed, collected, and then injected. You could see this is an embryo right here. This is the glass needle that's going to inject the DNA into the embryo. And then we're doing a surgical procedure. That's me right there. Somebody told me to smile for the camera. I mean, you do it anyway, even though it's ridiculous. And we're threading a catheter basically into the uterus of the pig in order to transfer those embryos in there. Now, these didn't end up turning into pigs used for xenotransplantation, but same process basically. Then you have to wait for about 4 months for piglets to be born. And then you take a tiny sample of DNA from them and you check to see if they got the edit that you wanted them to get. If they didn't, try again. Very expensive. So why is this important? We have a lot of people waiting for organs and we don't have very many donors. So this could solve a huge issue that humans face today. And yes, we can argue like, well, shouldn't we just be healthier? Shouldn't we just eat healthier and exercise more and not involve genetic engineering and animals and all that stuff? It's not always that. It's not always lifestyle choices that lead people to need organ transplantation. And this is from February 2024. This is how it could be used to solve the shortage. So just again, 2 months ago, how it could be used to solve the shortage. And maybe it's just a bridge. Maybe it's just a temporary transplant until a good match comes up on the organ transplant list. But if it lasts 2 months or 6 months and it gets you to a human donor, at least you didn't die waiting. March 21st, they did it. They took a genetically engineered pig kidney and put it in a human. And to the best of my knowledge, this man's still alive. But it took 69 genomic edits in that pig, 69 changes where they took out the pig-specific markers and put in human-specific markers. We could not have made those edits just by selective breeding or irradiating or using chemical mutagenesis. We had to use very targeted precision gene editing techniques like the CRISPR-Cas9 system to make very specific edits in very specific proteins so that this could even be possible. And like I said, that just happened. That was just in the news. Sounds great, right? Maybe you're thinking like, yeah, why don't we all do that? But there's always a reason. There's always excuses. There's always, I don't need it. It doesn't affect me. Why should I bother with that? And I want to remind you guys that we have privileges that allows us to have options and allows us to make choices. And not everybody has those choices. Not everybody has the ability to choose to eat something that is not genetically modified. Some people don't have a choice of what to eat at all. So you just have to eat what you eat. And if it comes down to having no food or having genetically modified food or genetically engineered food, you're going to go with the genetically engineered food. And that's not to say we should be forcing people who have no options to eat something that they don't want to eat. But it does mean that when we get to pick, just remember a lot of people don't get to pick. A lot of people don't have that choice. So do keep that in mind when it comes to thinking about genetic engineering as something that could have potential benefits, even if you opt out of it, right? But I guarantee you, if you ever end up on that organ donor transplant list, you'll take a pig heart if it's offered. Okay, thank you guys. Any questions? That's it.
I have a question.
Oh, we got a question online. Let's see, Kylie has a question.
Haley has a question. Hi, Lydia. Thanks for the great presentation.
Yeah.
I wanted to ask how long you think it will be until I can become resistant to the swine flu. That was not fun.
Oh no, I'm sorry. I'm not gonna
Yeah,
ask
I got it
medical
at
questions.
PAX when it was here a few years ago. We went to a
Oh.
concert afterwards and everyone at the concert got it.
I feel like in 2009 or something, I think I got a vaccine against swine flu. So there are some vaccinations available, but unfortunately, maybe that's in the future. I don't know, maybe 50 years from now, instead of getting a vaccine, we'll all just be gene edited, but it wouldn't necessarily be heritable unless you're gene editing eggs or sperm, right?
True.
So,
So not us.
so maybe if we got gene edited, then our offspring could be resistant to those things. So depending on, you know, who's running the study, who volunteers for the study, and are you, you know, going to be passing your genes on to the next generation? I mean, this is getting into some Gattaca territory if you haven't seen the movie. I mean, it's a great movie. Highly recommend that you watch it. So I don't know if I could answer that question. Vaccines are fairly effective and relatively cheap compared to gene editing individuals, I would say.
Yeah, I know there are some people who can't have vaccines, so maybe it's worth it for them, but
Absolutely.
not broadly.
Yeah, and that's the same thing where, you know, it comes down to like really custom medicine at that point.
Hmm.
Good question though. Okay. Anybody else?
If you're altering the DNA, wouldn't that automatically mean that that's going to be passed down genetically to us?
No, because we have two different types of our cells in our body. We have gametes and we have somatic cells. And our gametes are the ones that become eggs and sperm. Those are the ones that get passed down to the next generation. And then somatic cells are literally every other cell in our body, our heart, our brain, our lungs, our liver, our muscle, our kidneys, everything else in our body. Those pieces are what we usually target. We're not generally targeting the eggs and sperm, but in the case of doing genetic engineering in animals, yes, when we're doing microinjection and injecting an embryo, we're targeting a single-celled organism basically before it has the chance to divide into those other cells. So all the cells in the body, including the future gametes that embryo will eventually create when it makes its own sperm and eggs, will have that same mutation.
So it's potentially possible to do that in the future.
Potentially, but if you're talking about doing it for humans, you'd have to be looking at like, it's literally in vitro fertilization. You'd have to collect eggs, do in vitro fertilization with sperm in a petri dish, do the injection, and then transplant that embryo back into the human, have that human grow up a baby, and then that baby would be born with the modified gene, whatever that gene is. And that might be how we treat debilitating, like lethal mutations in the future.
Down syndrome, for example.
Down syndrome's a little bit more tricky because that is actually because you have one extra chromosome and it's not just a single gene, it's like a whole extra huge piece of DNA. But if you have single genes and you know, like my partner has it, I have it, we do genetic testing right now on human embryos for that reason. So when people are doing IVF, you'll genetically test the embryo before transferring it, just to make sure you're not using an embryo that you know is going to have some sort of lethal mutation. But we're not quite at the point where, okay, we've identified the lethal mutation and let's go in and fix it. And then we're not quite there yet. Maybe in some countries, but not in the United States. We're not there yet. Awesome. Okay, any other questions? Anything on Zoom? Okay, I think that's it. I don't see any questions in the chat. So thanks again for coming by, guys. I hope you found it interesting. And yeah, I didn't even get to talk about poop yet. So, you know, that's like all the rest of the slides. But you know, if you want to talk about inflammatory bowel disease or infectious E. coli, I'm happy to continue talking about it, but I will let you guys go.
Thank you. Thank you very much.
Bye. Thanks, Amal. Oh yeah, if you didn't grab the sign-in sheet, please do. That'd be fantastic.
I'll meet you there in 1 minute.
Thanks, guys.